Good morning. Can you guys hear me in the back? I can't tell if this is like projecting. Okay, great. Thumbs up. All right. My name is Drew Williamson. I just joined the faculty at Emory after completing a postdoc with Faisal Mahmood at Brigham and Women's Hospital, where our focus was on deep learning applied to all sorts of pathology data, usually histology images. Those are like microscope images that pathologists use to make diagnoses, but also often genomic data, natural language, radiology data, sort of everything in between. But today I want to talk specifically about histology images and the features in them. And so when I was going through pathology residency, let me advance to the next slide. There we go. When I was going through pathology residency, I would often see images like this. So this is a core needle biopsy of a patient's breast mass. And, after marveling at how beautiful it is and how amazing it is to see the cells and the nuclei and examine their morphology, an annoying attending would often ask me to describe what I'm seeing down the microscope. And this is like kind of the core way, one of the core ways you learn pathology and how to examine pathology slides. And so I'd have to give some morphologic description of the histologic features, say, so the like core characteristics that might communicate to my attending or to another pathologist what I'm looking at here. So I could say something about the tumor cells. Can you see my mouse? Nope. You can see like the purplier bits are arranged into these circles, and that's kind of characteristic of a breast cancer. And this idea of extracting histologic features or features from histology images has a very long history. And if we go back and look at the first morphologic description of cancer cells, this is an English translation of a German paper from 1838. So this is like pre-H&E era, which is what we use in pathology nowadays to examine slides. We can see basically, of course, this description of these histologic features. Now, back in 1838 and 1840, we weren't using the same jargon and terminology that pathologists use today in 2024. But, reading some of these passages, you get a sense that someone's looking down a microscope at some cancer, which up until this point had only been described via its like gross image, gross as in like macroscopic features. You can see these minute globular cells which form the real Seminium morbi in several forms of carcinoma, et cetera, et cetera. So, essentially immediately we have these kind of natural language histologic features. And other descriptions, like a cell when viewed under the microscope appears to contain corpuscles in its interior. I'm not sure what that means. Maybe it's referring to like the nuclei or the nucleoli of the cells, or other cells may have other little bits and pieces in them. But also, essentially immediately with these natural language histologic features, we also get these quantitative features that actually match pretty well the sizes that we would expect, based on looking through our microscopes now in the 2020s. So we basically right away have these two paradigms of histologic features, the quantitative features and the sort of natural language histology features. And really in the 1920s, this idea of morphometry or the quantification of these histologic features really took off. So we have like the first description, quantitative description of how cells double in size when they're undergoing cell division. Makes sense. And then in 1929, cancer nuclei are bigger than normal cell nuclei, which is something both of these, we recognize as true today. They've held up to the test of time. And probably my favorite publication from this era has this great chart that I think, with a little cleaning up, wouldn't look too out of place in a journal article published today. This is a really quantitative and comprehensive description of how the nucleoli of cancer cells tend to be bigger than those of normal cells. And, I love these distributions that Eva Hammider from the Mayo Clinic drew for us. And this was profiled, this was actually presented first as a talk at the AACR annual meeting a couple of years before, and I love this passage. Now, Dr. McCarty and Hammider, they conclude that the cancer cell has at least one differential characteristic, and that this must be used by those who expect to reduce the mortality of cancer. And so no, I don't think anyone's targeting, for example, the size of nucleoli in cancer cells today for cancer therapy, but Fortunately for cancer patients, we've identified a lot more differential characteristics of cancer cells that we can use to reduce cancer mortality. Maybe some genomic features like presence of targetable mutations, things like that. And, if we look at how pathology is taught today, it's really still this combination of morphologic descriptions via natural language and text and quantification. So there in the top right, we have this table for gastrointestinal stromal tumors that has some cutoffs based on the size of the tumor or the mitotic rate of the tumor that tells us something about how that cancer is going to behave. And then we have these nice morphologic descriptions that I'm going through residency and I'm reading these and picturing histologic images like those on the right in my head and trying to match those up with what I see on the slide to make a diagnosis. And often in these textbooks, you'll have this perfect correlation between a histologic image and some description of what the author is trying to show you from that image. And really, if we think about extracting histologic features in a very manual way today, that's basically what the practice of pathology is. So we'll look at a histology image, maybe at high magnification and low magnification. We'll zoom in. Some practices will write a microscopic description in natural language that describes exactly what they saw under the microscope. And that's to communicate to other pathologists who may review those slides down the line or treating clinicians. And then we'll give maybe some more structured text for a final diagnosis and say, this was a partial gastrectomy from a patient, and we're looking at a gastrointestinal stromal tumor that happens to be epithelial type, and then maybe give some quantified features, et cetera, et cetera. But, we're at this bioinformatics conference, and I think what we all really care about is how can we do this in an automated fashion. And today in 2024, we have a couple different options for using AI more generally to do this extraction of histologic features and have computationally extract, what is important in these histology images. And really, there's been two main paradigms now to do this computationally. We can extract human interpretable features that might be something like the average nuclear area of the tumor cells, or maybe we extract the tumor region, we get the fractal dimension of that, or we can extract these deep learning-based features that bear no relation to human interpretable features. Let's start with the human interpretable features. This is a figure from like a prototypical work extracting these human interpretable features. This is from some folks at the PathAI company in Boston. And what they did is they trained some deep learning models to identify various types of cells in a histology image and various tissue types. And then once they had those cells and tissue types segmented via some deep learning methods that we won't talk about, then they extract these human interpretable features. That might be the fractal dimension of the cancer-associated stroma, the eccentricity of largest region of necrosis, et cetera, including some human interpretable features based on the tissue regions, like the stroma or the tumor, and also some cell-based features that might be, the eccentricity of the nucleus and things like that. And then they go on to show that these human interpretable features have some correlation with genomic features, which makes sense. Back in the 1920s, we knew that the cell size doubles when the cell's dividing. Nowadays, we know some molecular markers for cell division, and so I think it makes a lot of sense that there'd be some correlation between these human interpretable features, such as, for example, cell size and molecular markers. Now, another kind of prototypical work in this field is this paper from Anant Matabushi's group, who's now at Emory, where we detect some human interpretable features, such as the presence of mitosis, the cancer nuclei, or maybe the tubules in a prostate cancer or breast cancer or something like that. And then we go through the histology images, we find those features, we quantify them, we get the distribution of those features, maybe across images. And then in this case, they show some differences in survival based on the quantification of those features. So if you really love this idea of human interpretable features, there's a couple of different options for you nowadays to go ahead and do this. Histomics TK or Histomics Toolkit, published by the Digital Slide Archive, is probably the most comprehensive resource for doing this sort of thing nowadays. So they have a very nice GitHub that will allow you to go ahead and extract features in addition to segmenting nuclei and tissue regions and that sort of thing. There's a bunch more tools that do like nuclei feature extraction, but then you have to define or nuclei segmentation, but then you need to go ahead and define your human interpretable features yourself and extract them. And there's a couple different options, new and old there. I'll say HoverNet is probably like the standard that people use these days, but recently there's been a couple of publications showing better performance for this nuclear segmentation and classification of nuclei into, say, tumor, non-tumor, stroma, lymphocyte, that sort of thing. All right, next let's talk about these deep learning-based features. So again, these features are just going to be vectors or matrices of real numbers. They bear no relation to anything that humans can interpret. We don't know what any individual feature means, but we know that comprehensively they're very expressive about what's going on in these images. We just need some further deep learning based tool to work on top of those features to tell us to do some task based on those features. And so this is like the prototypical work here, where kind of a seminal work in computational pathology by Cordray et al. In Nature Medicine back in 2018, where we take some images from the TCGA, we separate them into train, validation, and test. We take those big images, we split them up into little patches, and then for each patch, we extract some deep learning-based features. In this case, using a pre-trained Inception v3 models that's been pre-trained on natural images, and we're making the assumption that features, a model that's trained on natural images, so pictures that we see all around us, will be expressive, at least to a certain extent, from histology images. And then you aggregate all those features together from all your patches and do some downstream computation. And here's another sort of work, another similar work from my group in Boston, doing a similar thing. We extract some deep learning-based features from little image patches, and here we're using ImageNet, or another ImageNet pre-trained network to do that feature extraction. And then we do some computation on top of it. So, transfer learning was incredibly important to the development of deep learning-based computational pathology, just because back in 2018, 2019, 2020, we really didn't have access to enough pathology slides to train a model to extract very expressive histology features and have that training only be on histology slides. So we needed to rely on very large databases of images of any type, usually ImageNet or similar databases like that, to train these feature extraction. But now in 2023 and 2024, there's been an explosion in computational pathology of foundation models. And by foundation model, I mean the same thing that I mean when I talk about foundation models outside of pathology. It's a model trained on a very large corpus of data that is very good at extracting features from individual pieces of data, and then you can use those features for a bunch of downstream tasks. And in the pathology world, there's been quite a few publications and preprints about these foundation models now that people have been scanning slides routinely for 5 or 10 years, and we have enough histology slides to train a feature extractor only on histology slides. So my group in Boston published one called UNI, and here's just a demonstration of the various downstream tasks you can do with these. But the key point that I want to draw your attention to is that for every downstream task, we start with the same recipe. We take a histology image, we apply UNI, we extract those deep learning-based features, and then we can train another model to do something with those extracted features, be it a classification task on those patches or a whole slide. We can do retrieval if you want to do some image search, et cetera, et cetera. The world's your oyster. And just for completeness sake, we have this table or this figure in our publication. And, back in 2018, we were using ResNet 50 pre-trained networks, and those perform significantly worse than these foundation models that we can train entirely on histology images. And if you love this idea of deep learning-based features, there's a couple different options. Here's some publicly available ones that you can go download the model and check out these GitHubs and download the model and feed in your images and get features straight away. And then decide whatever you want to do for your downstream task based on those features. And of course, I'm a little biased. I think UNI is great, and we have some explainer Jupyter notebooks linked from our GitHub that you can take a look and play around with those features. And then our model itself is available on Hugging Face, and you can go and download the fully trained model and do whatever you want with it. Okay, so there's our like two main paradigms of computational histology feature extraction, and there's some trade-offs between these two. I think the human interpretable features, we're going to define them before we do anything downstream, and they have a very clear meaning, but we're injecting some purposeful bias when we decide what features we're going to extract. I know that the size of the nucleus is important for determining cancer versus non-cancer, and so I'm going to have nuclear size be one of my human interpretable features. And because of this purposeful bias, and I'm defining them ahead of time, they're easy to connect to biology. I know the nuclear size is related to cell division, so of course it's going to be easy to interpret that through the lens of biology. The deep features are kind of the total opposite. We're not injecting any bias about these features ahead of time, only defining the number of features that we want the model to extract. And that's, you're sort of doing this trade-off between bias, let's say. We're not biasing the model in terms of which features to extract, which means it can detect things that I, as a pathologist, didn't know that I should be looking for looking at these images. The counterpoint to that is that it may pick up things that I don't want it to look for. So there's been a number of works in computational pathology showing that these models, even though they're trained on histology images, can be biased by demographic factors like self-reported race or socioeconomic status or something like that. So there may be some information in these histology slides that we don't want the model to pick up on, that it's going to do so anyway. So for example, the TCGA aggregates slides from all over the United States. Those slides get scanned on different scanners, and the signature of those scanners may be hidden in these images. And similarly, the histology slides themselves are made at different institutions that have different concentrations of the stains that we use to actually make the slides. And because those institutions serve different patient populations with different racial makeups, you can imagine that a model that picks up on those differences in staining may be biased by race in that way. Now, if all you care about is performance and you don't care about anything else, I think your best option is to go with these deep learning-based features. They tend to have higher performance, but again, could be brittle to other things. And here's this paradigm, but I kind of find this paradigm as a pathologist to be deeply unsatisfying, because neither of these is really what I do when I look at an image. Like maybe I do extract some deep natural neural network based features in my mind implicitly, but I certainly don't think about the average nuclear area. I'm not measuring nuclei when I look at a slide, or considering the exact density of lymphocytes or something like that. And if we go look at a microscopic description of a slide, we have all this jargon. And this jargon, I think, makes pathology text very difficult for people outside the field to read. But for me, it's symbolic of the almost 200 years of people looking down microscopes and putting literal blood, sweat, and tears into figuring out what's important in these images. And pathologists know that terms like myxoid stroma and mucosa and epithelioid have an implication for not only what they see on the slide, but whatever is happening downstream. So like the behavior of the entity that we're looking at on the slide. And these jargon sort of things is how we learn pathology. And so this is a wonderful textbook that I've made very small so you can't read it, but it's called Quick Reference Handbook for Surgical Pathologists, that has this wonderful section where it goes through the jargon and shows the term on the left there, a definition of that term, and a histology image with that demonstration of that term. And that's great because some of these terms are kind of outdated. These are back from the 1800s. We started using words like festoon-like. And pathologists, we love our food analogies. So the term coffee bean nuclei has a distinct meaning of pathologists and sort of conjures a couple different entities into our minds. And what I really want is kind of like a pathologist feature extraction, where I want a model that uses the language that I already know, and so that would make it easy for me to interpret, but also I think uses these terms that we've shown over 200 years to be important in these images. And in the past 6 months to a year, I think we've taken a step towards doing this kind of pathologist feature extraction. And there's 2 papers in Nature Medicine, one from my group in Boston, that sort of takes the first crack at doing this sort of thing. And both these papers are very similar. We just get our data from different places. And so the top paper from James Dowd's group at Stanford uses Twitter, or I guess maybe it's called X today, to get paired image and text data. And we use pathology textbooks and PubMed articles. And both of these papers use a similar model and a similar training paradigm to train a model that understands both natural language and histology images. And so we're going to have 2 different encoders, one encoder for each of the 2 modalities. So we're going to have a text-based encoder that's going to be pre-trained on a large corpus of text, and an image encoder that's pre-trained on a large corpus of histology images. And the way we train this model to understand both of these modalities, that we take a paired image and text, so maybe a tweet with a histology image and a pathologist caption. And we put the caption into the text encoder, the histology image into the image encoder, and we project them both into a shared latent space. So we extract images with the same number of dimensions, or extract features, the same number of features from both modalities into the shared latent space. And then we train by saying, if the image and the text came from the same tweet or the same caption in a figure textbook, we want those features to end up in the same or a very similar place in the latent space. And if I take a tweet about a different tweet, or a tweet with an image, one tweet with an image and a different tweet with a caption that doesn't describe that image, I want those 2 things to end up in a very different place in this shared latent space. And it turns out, if you do this over a large corpus of data from Twitter or PubMed or in journal articles, you can get a model that understands both of these and is very good at taking an unseen histology image and mapping it to a place that's expressive in this shared latent space. And then what do you do with that? So you can do something like image retrieval. Say we have a fixed histology image, and then we map that into the latent space, and then we generate a bunch of descriptions that all live in that latent space. Then we go pick the description that's closest to that image in the latent space and project it back into text. And we can generate some text like that. And we can do classification of different, for example, patterns of lung adenocarcinoma by just taking the histology image and a single word description, micropapilla, acinar, etc., and figuring out which of those single word descriptions is closest to the histology image in that latent space. And as I mentioned, we can do a kind of generative version of this. So we take an image and we project it into the latent space, and then we go from where it ends up in the latent space and we generate some text based on that. And in that method, using that method, we can generate captions for histology images. So here's a couple cherry-picked examples that look pretty good of doing this, and where at least the model is getting the right diagnosis and maybe some description of what's going on in that image correctly. Another somewhat cherry-picked example, this model doesn't get everything correct, but when it does get things incorrect, they tend to be mistakes that are reasonable to me as a pathologist, and mistakes that I would make as like a medical student or a first-year pathology resident. So, this is an image on the left of a cardiac rhabdomyoma. The model is saying it's a clear cell carcinoma or fat necrosis, and on the right I have a clear cell carcinoma on the top and fat necrosis on the bottom. And, I think to me those look kind of similar to this image on the top. So kind of a realistic mistake. And, basically this is where we're at with these papers. We can take a corpus of pathology images, we can run it through either of these 2 models, and for each image we get some natural language description of what's in that image. We extract the sort of natural language histology features there. And if you're excited about there's some GitHubs that you can go. Again, our model, the conch model, which is ours, is publicly available. You can download the model from Hugging Face and start extracting these natural language features, from your own histology images. So really in 2024, we're at this kind of paradigm. We have like 3 different options. We have our pathologist features, in this case, some natural language descriptions. I think, if I can go back here, one thing you could do to make these more structured is take these text descriptions and run them through GPT-4 or any other publicly available large language model and have it use another AI model to extract some more structured text. So we have our pathologist-based features that we're kind of coming to the forefront now. We've got our human interpretable features and our deep features. And really, if you want to extract histology features, you're probably going to be using one of these three paradigms. And bring it full circle, going back to the very first descriptions of cancer cells. We have these old, very, very old histology features, which is kind of what we're reinventing now, just in automated fashion. A natural language description of corpuscles in the interior of a cell or some measurements of the size. I will leave you, since my time is up, with this quote from that 1840 paper. We think that these observations will put an end to the confident anticipations expressed by some pathologists and entertained by many persons in the profession that some simple microscopic test of malignant growths would be discovered. I think, thankfully, we know that pathologists are that simple microscopic tests that can distinguish between normal tissue and malignant growths. That's it for me. Just started a lab at Emory. I'm recruiting at all levels. If you're looking for a position extracting histologic features and doing other cool things, please reach out. Thank you. Yes.
Michael from Virginia University. It's a great talk. So this is critical algorithms to provide a natural language description of image, right? So would you elaborate how to evaluate the accuracy of the natural language description?
Yeah, great question. It's in a word difficult. I think, in our paper, we took a couple different approaches. If you're just extracting these sort of captions, kind of the only way to do it is to have pathologists look at the image and the caption and decide this is good or bad. I think some natural questions arise of like, are you going to just have like a binary good or bad, or are you going to have a pathologist rate it? And pathologists are busy and expensive, and so it's difficult to do that for a very large corpus of data that you want to check. Other things we did was have like multiple choice questions that the model would have to answer and say, given a couple different options, which diagnosis is closest to this image or caption, that sort of thing. And then, another thing you can do is just use these features, just extract the features and then use the natural language features for like downstream tasks and say, let's say we want to predict whether an image shows cancer or not cancer. If we use the natural language as input to a downstream model to do that, is the natural language that we extract better for this downstream task than another model or something else? But it's tough. Of course, like if you're OpenAI or some other multi-billion-dollar company, you just get like thousands or tens of thousands of humans to like rate your responses and upvote them and downvote them, et cetera, et cetera. But hard to do that as an academic lab. Yes.
Are all pathologists becoming this computationally focused? And if not, how are the kind of others reacting to these changes in the field?
Yeah, I think pathology and medicine in general is very slow to change. I think pathologists even slower than the rest of medicine. I would say, and pathology is like a very old, in terms of like the age of the people practicing pathology is like older than most other medical specialties as well. So there's like a lot of inertia to bringing these computational approaches into pathology, not the least of which is that though these things perform well on paper or in papers, I'll say, they've been not very well tested, if at all, in actual clinical practice. And so there's that inertia coupled with some skepticism about computational approaches in general and AI and the fear and the hype around it. And I think anecdotally, when I've been involved in like residency recruitment, I've seen basically every year I've been involved in more and more people expressing an interest even before they start residency in computational things. And when I started, I was like the only person that was interested in computation and histology images. But 4 years later, there's like 5 or 6 people every year who say, like, from the start, this is what I want to do. And I think part of that is just like the mainstream hype around AI, like bleeding into medicine. And then people who are in their undergrads are majoring in computer science or math or something like that, that have some more familiarity and know how to interpret these things.